Welcome to TNP and Expert Education. In this video, we are going to see about somatic hybridization. For somatic hybridization process, the protoplast must be isolated. So first, we are going to see about protoplast isolation. Protoplast are spherical, nacked plant cells obtained by the removal of cell wall. Simply, protoplast is called a cell without a cellulosic wall. Protoplasts are surrounded by plasma membrane and potentially capable of cell wall regeneration, growth and division. The protoplasts are excellent tool for synthesis of novel combination of the genes and are essential part of the overall processes required for the genetic engineering of the plants. Isolation of protoplasts. There are two methods are available, mechanical method and enzymatic method. Let us see one by one. Mechanical method. This method is suitable for isolation of protoplasts from highly vacuolated cells of storage tissues. For example, onion bulb, radish root, etc. See this picture. The researcher actually find out the protoplast and isolated the protoplast from the cell. So the cells are kept in the suitable plasmolyticum and protoplasts are isolated by cutting the plasmolysed tissue with a sharp blade so that protoplasts are released from the cells through the cell wall when the tissue is again deplasmolized. See the picture how the researcher cut the cell with a sharp blade and how the protoplasts are isolated or separated from the cell wall. The H is actually protoplast isolated from the cell wall. So this is about mechanical method. Advantages of this mechanical method: it eliminates the unknown effect of enzymes on protoplast. But it has some limitations. Only a small number of the protoplasts can be isolated. Method is very tedious and time consuming process. Not useful for isolating protoplasts from meristematic tissues and mature and less vacuolated cells, because the meristematic tissues are very minute tissues. So it is difficult to find out the protoplast and cut with a sharp blade. Viability of the protoplast is very low because of the presence of substances released by the damaged cells. Enzymatic method. Cocking 1960 demonstrated the possibility of enzymatic isolation of protoplasts from higher plants. Jacob et al. 1968 for the first time employed commercial enzyme preparation for isolation of protoplasts and subsequently regenerated the plants in 1971. By using this method, protoplasts can be isolated from any part of the plant body, but it is easier to isolate protoplasts from the mesophyll of leaf and pollen, mother cells or tetrads. So here are commonly available enzymes. Here we are giving enzymes and where the enzymes are isolated, the source of the organism. See that cellulases, Monosuca or S, cellulase or 10, they are isolated from Trichoderma viride, the source of the organism. Hemicellulase, Rozyme HP150, isolated from Aspergillus niger. Pectinase, Macerozyme O10, Macerase, isolated from Rhizopus species. Actually, in enzymatic method of protoplast isolation, there are two methods. First one is the two-step or sequential method. The tissue is first treated with macerozyme or pectinase, the enzyme which is separate the cell by degrading the middle lamella. Actually, the cell wall consists of the pectin and also cellulose. So, in this two-step method, one by one, the cell wall is degraded here. So, the enzyme is added here is pectinase, which degrade the middle lamella. Those free cells are then treated with cellulase. ACE is actually enzyme. When the word mostly complete with the ACE sentence, that is called enzyme. cellulose, that is carbohydrate compound. This is cellulase, which releases the protoplast. Cellulase enzyme digests the cellulose present in the plant cell wall, while pectinase enzyme break down the pectin, holding the cells together. So this is about two-step method. And this is one-step method or simultaneous method, because here the two enzymes are added at a time in the mixture of the cell. The tissue is subjected to a mixture of enzymes in the first step reaction, which includes both macerozyme and also the cellulase. So in this step, the method is generally used because it is less labor intensive, while the yield of protoplasts isolated by two-step method are better for culture studies. Advantages. When compared to the mechanical method, in this enzymatic method, large quantities of the protoplast can be isolated. Shrinkage is very minimum and the deleterious effects of excessive plasmolysis are minimized here. Cells remain intact and are not injured as observed in the mechanical method. Because in this mechanical method, the researchers cut the cell with a sharp blade, but here only we are adding the enzymes. So the injured is minimized here. Purification of the protoplast. After the material has been incubated in enzyme solution for an adequate period, the incubation vessel is gently swirled, or the leaf pieces are gently squeezed to release the protoplast held in the original tissues. Enzymatically digested mixture would contain all the compounds like subcellular debris, undigested cells and also digested cells, broken protoplast, besides intact and healthy protoplast. So I have to purify and separate the protoplast from this mixture. This mixture is purified by the filtration, centrifugation and washing method. The large debris is removed by passing through a metal or nylon filter. About the size is 30 to 100 micrometer and further purified by centrifugation process. See that picture? First they filter out the suspensions and then centrifuge the suspension and last washing. So this is called purification process. The next process is to check the viability of the protoplast, because for further studies we are only taking the viable cells of the protoplast. Observation of cyclosis. Cyclosis means cytoplasmic streaming, that means the movement of the cytoplasm is an indication of the active metabolism. Oxygen uptake measured by an oxygen electrode, which indicates the respiratory metabolism. And we have to check the photosynthetic activity of the protoplast. Exclusion of non-viable cells by Evans blue dye technique. Viable cells actually find out by staining with fluorescent diacetate technique. So in this method, the only live cells, means viable protoplasts, emit the fluorescent color. The next process is the culture of protoplast. The first step in the protoplast culture is the development of the cell wall around the membrane of isolated protoplast. So this is followed by induction of the division in the protoplast derived new cell, giving rise to a small cell colony. By manipulation of the nutritional and physiological conditions in the nutrient media, the cell colonies may be induced to grow as a callus continuously. They are regenerated as a whole plant. So under the process of de-differentiation or redifferentiation process, the callus which will form the whole plant here. So this is about culture of protoplast. But for somatic hybridization, after isolation we go for the next process. Just see that. Protoplast fusion or somatic hybridization. So this technique, actually somatic hybridization, means we are collecting the cells from the somatic cell. So this is called somatic hybridization. This is one of the most important uses of the protoplast culture. This is particularly significant for hybridization between a species or a genera, which cannot be made to cross by conventional method of the sexual hybridization. So this is a very important thing. We have here we are fusing many genera and also the species. Somatic hybridization means in vitro fusion of plant protoplasts derived either from somatic cell or somatic plant or from two genetically different plant. is called somatic hybridization. So how to fuse the cells? What are the methods of protoplast fusion? Let us see one by one. Spontaneous fusion and induced fusion. So we are going to know about the spontaneous or induced fusion. This is the overall picture of the protoplast fusion. You just see the picture. How to collect the explant A, explant B. Then here they are selected the leaf tissue, sterilized leaf tissues from explant A and explant B. Then after they further go for cell wall removal by cellulase enzyme reaction. That means protoplast collection, isolation process. And the next process is fusion, protoplast fusion of two different explant A and explant B. And the result they are getting callus, fused callus, in hybrid callus. It mixed with explant A and explant B, and through by the tissue culture process only. And after that, from the callus, here the shoots are regenerated, and then plantlets, complete plantlets are formed. So this plantlet actually consists of the characters of the explant A and explant B. So this is actually called a somatic hybrid. Okay, let us see the methods of fusion. Spontaneous fusion. The protoplasts during isolation are often fuse spontaneously, and this is a natural mechanism, a natural phenomenon. This is called spontaneous fusion. Simply, the physical contact is sufficient to bring out the spontaneous fusion among similar parental protoplasts. The occurrence of multinucleate fusion bodies is common when the cells are prepared from actively dividing cells. So the spontaneous fusion usually gives rise to the homokaryon only. Homokaryon means similar characters of the plants. Plant A, explant A means explant A. It only gives the homokaryon, not the heterokaryon, not the hybrid. So maximum the spontaneous fusion result is a homokaryon. This is induced fusion, chemofusion. Because the chemicals used here to induce the fusion, it requires fusagen. The fusagen first brings the protoplasts together and then causes them to adhere to one to the another. So automatically it will fuse together. The first one is sodium nitrate. The protoplasts are suspended in the aggregation mixture of 5.5% sodium nitrate in 10% sucrose solution. And second one is the polyethylene glycol, PEG, shortly called PEG. And this is one of the main important fusagen used in the protoplast fusion, most probably widely it is used. So polyethylene glycol, don't forget that 1 ml of protoplast suspended in a culture medium with 1 ml, 1:1 combination, 50% of the PEG, and the tube shaken for only 5 seconds. The protoplasts are allowed to sediment for 10 minutes and washed with the growth medium and examined for successful agglutination and fusion. And third one is the calcium ions. Here the protoplast fusion is by calcium ions, 0.05 molar calcium chloride mixed with 0.4 molar mannitol. Mannitol actually here acts as a osmotic regulator. So for it bringing about the fusion of the protoplast. So mechanical fusion. The isolated protoplasts are brought into intimate physical contact mechanically under the microscope using the micromanipulator and perfusion micropipette. And the next one is the electric fusion. Here simply the electric current is supplied. The protoplasts are placed in the small vessel. You see the picture? It containing the electrodes and a potential difference is applied. Then the protoplast will line up between the electrodes. See that? First it will line up between the electrodes. And afterwards the extremely short square wave electric shock is applied. The protoplast can be induced to fuse. So this is about electric fusion. Okay, after fusion technique, we have to select the somatic hybrids. Selection of the somatic hybrids. The protoplast suspensions recovered after a treatment with the fusagens consist of following types. Actually, the protoplasts are maybe unfused protoplast or fused protoplast are the products of the homokaryon. Homokaryon means the same species may be fused together. And heterokaryon. Heterokaryon means hybrid protoplast. They are produced by fusion between protoplasts of two different species. So that is heterokaryon. Homokaryon, same species. So effective strategy has to be employed for the identification of the somatic hybrids of heterokaryon in nature, and they should be isolated. So this step is called somatic selection of the somatic hybrid cells. So these are all the methods of selection of somatic hybrids. So first process is the somatic visual markers. Visual markers means even the explant A have some different colors of callus, explant B has some different colors of callus. So by visually we have to select that whether it is a hybrid, somatic hybrid or not. So this is somatic visual markers. And biochemical basis for complementation. That means acts in autotrophy. Actually, the parental protoplast of, for example, Nicotiana glauca and Nicotiana longstophy requires an axin compound in order to proliferate. But whereas the hybrid callus tissue, it needs no such requirement, because each and every process of the callus formation we have to add axin. But here the hybrid callus, it has axin autotrophy. That means without the axin it will grow. So here this is axin autotrophic. The hybrid only grow without the axin. This is biochemical basis for complementation. To culture the entire protoplast population, and the next process is the labeling. Protoplast of two parents may be labeled by different fluorescent agents, which will then enable the selection of the hybrids. And another one technique is fluorescent assorted cell sorter, FACS method. So this method also used for the selection of the somatic hybrids. Then symmetric hybrids and asymmetric hybrids. There are two types of hybrids only formed. Some somatic hybrid plants retain the full or nearly full somatic complements of the two parental species. They are actually called symmetric hybrids. But asymmetric hybrid means many somatic hybrids exhibit the full somatic complement of one parental species are lost during the preceding mitotic divisions. Such hybrids are referred as asymmetric hybrids. Okay. Okay, if you like this video, like, share, comment and subscribe this channel. Thank you.